Good
Good
morning, sir.
morning. Good morning. So, today we will continue from the previous class. I think previous class towards the end, there was a problem of internet, so we had to stop it. I think before completion, it was. We were in the previous class. We have finished that xylem tissue. No, do you remember? Hello, hello.
Yes, yes, sir.
But in the previous class, up to what portion did I teach? I think Xylem was complete, no?
Yes,
Xylem
sir. Till up till vessel.
up till vessels.
Yes,
Okay. So there is so in the xylem tissue there is tracheids and vessels. Is it tracheids and vessels were there? Vessels are made of that vessels are tube-like structure made of by joining many cells end to end and form a tube-like structure. So they are these tracheids and vessels. They are the main transporting uh, uh, transporting elements. So and beside this xylem, the tracheids and vessels, there is xylem parenchyma. Okay, xylem parenchyma here. Xylem parenchyma is there, and also xylem fiber. Xylem parenchyma and xylem fiber. Xylem parenchyma is the ordinary, you can say the parenchyma tissue, that living tissue, living cells. Okay, and this parenchyma, they store food material in the xylem tissue, storage for food and short distance transport, lateral transport. And this xylem fiber, these are the sclerenchyma fiber actually present in xylem tissue. Their xylem fibers are nothing but sclerenchyma fibers. Okay, they are very long, and so this xylem sclerenchyma fiber, as we know, they are dead, is it? So what they do, they provide mechanical strength, additional mechanical strength they provide. Otherwise, they don't have. So this xylem tissue become very strong. So, and these actually xylem phloem, they are mainly for the long distance translocation. And if they are not very strong, then there will be problem. If they are damaged easily, then there will be problem for the plant. Okay. Another thing that xylem, this xylem tissues are also called that there is a type that there is a primary xylem and a secondary xylem. Primary and secondary. What is this primary? Primary xylem is during the development or the growth of the plant, the xylem tissue which appear first. Okay, and the secondary one is the one which appear secondary xylem appear later on. These are in the later stage of development. Okay, one is another thing is that is we call the xylem is protoxylem, and xylem is metaxylem. This protoxylem and metaxylem, they are the type of primary xylem. Primary xylem is which is formed first during that from the procambium. This primary xylem, they are formed from the procambium, and in the early stage of the development from the primary meristem, and primary meristem of the complex tissue is called the procambium. Okay, this, so the primary xylem is protoxylem and metaxylem. Protoxylem, this one formed first, and when it is formed first, naturally the cells are small. The component cells are they small, and their amount, total quantity is less. Okay, metaxylem, which is formed later on, this is metaxylem, and their quantity, usually the cells large, that is the element component cell, so they are large, and quantity is more in quantity. So protoxylem and metaxylem. This arrangement of the protoxylem and metaxylem is slightly different in case of root and stem. In case of say this is protoxylem, metaxylem first formed. One term is there, endarch and exarch. Endarch exarch condition is say xylem may be endarch and xylem may be exarch. Endarch condition or exarch condition, it is the relative position of the protoxylem and metaxylem. In the endarch condition, protoxylem is inner and metaxylem is outer, means on the inner side protoxylem and on the outer side metaxylem. And in exarch condition, this is a reverse of the endarch. In the exarch condition, protoxylem is on outer side or on the outer face, and metaxylem on inner face. Okay, so this condition, this enlarged condition, is characteristics of a stem. Okay, in a stem it is enlarged, and exarch condition, it is the characteristics of roots. In the roots, protoxylem on the outer side and metaxylem on inner side. This thing we can see by drawing. Say if I draw a diagram, say if I draw one vascular bundle like this, then on this side, this is protoxylem, and this side is metaxylem. Okay, so this one is enlarged type. This is found in the stem. In case of root, what happened? The arrangement is just reverse. Less, this is protoxylem, and this is meta. So this condition is exarch. Exarch condition is characteristics of root. Okay. One thing about that I have told that there is primary and primary xylem and the secondary xylem. Primary xylem, that is appear of course early in the stage. Here, this one, primary xylem appear first, all right, and this one is from the primary meristem. First, from the primary meristem. Let me from primary meristem, this one is formed, and the secondary xylem, this is produced from the later stage of growth, from the secondary meristem. Okay, so primary, this is in the earliest stage of the development when the primary body is formed. This secondary xylem is produced only in case of gymnosperm and in dicotyledonous plant. Okay, in case of monocot, whole content is a primary xylem. There is no secondary xylem because in monocot there is no lateral meristem, and that's why there is no secondary growth, and so secondary xylem is absent. So secondary xylem is present in dicot and gymnosperm. This is secondary meristem is fascicular and during their secondary growth. This secondary xylem is produced during secondary growth, and that takes place from the secondary meristem. So primary xylem is inarch in the stem and exarch in the root. Xylem translocate. Their main function is translocating water and mineral. This is a long distance translocation. Okay, let us come to the next complex tissue is the phloem. Phloem is for the translocation of organic food. This is for the translocation of organic food. In case of phloem, phloem is made of in the phloem there is components are that is seed tube, companion cells, phloem parenchyma, and phloem fibers. Okay, here is say this is a diagram of the phloem tissues showing all the component of the phloem. Main component is the seed tube. Main component is seed tubes, and these tubes are very long tube-like structure made of many cells placed end to end. This is one of the cell, this one is one of the cell, next cell is this one, next this side one. So this way there is a tube-like structure is formed. This seed tube is a living thin walled. There, say the seed tube is made of seed tube elements. Seed tube is made of seed tube elements, means component cells. Okay, and they are placed end to end. All right, they are thin walled, living and contain cytoplasm also. And the special thing in the seed tube element and in the seed tube is that this end plate, here you can see this plate, that is the end wall. The end wall is porous. There are holes, and this is known as this end wall with pores, it is known as seep plate. So this is this. Here it is showing the seep pores. These are holes. Okay, small holes are there. Here it will be clear. So holes are there. These are the seep pores, and this whole plate, this one is known as seep plate. This tissue is, this cell is living, but one thing is that when the seep tube is mature, in them there is no nucleus. You have to remember this condition, that living cell without nucleus in plant is the seep tube element. Okay, and as the nucleus is not there, so their activity is controlled by the closely associated cell. These are known as companion cell. Seep tube is associated with a special living parenchymatous cell. This is known as companion cell. They are not easily detachable from the seep tubes, and they have dense cytoplasm and prominent nucleus. So they can regulate the activity or turgidity of the seep tube. So here the companion cell, they are specialized parenchyma cells which are closely associated with seep tube element. Seep tube elements and companion cells are connected by peat fields. Peat fields means there is a plasmodesmata connection between these two cells. So peat field present between the common longitudinal wall. Companion cells help in maintaining pressure gradient in the seep tube. This is important point to remember for objective question to answer. Then there is this important point is this companion cell. Seep tubes and companion cells are absent in case of gymnosperm. Okay, seep tube and companion cells are characteristics of angiosperm only. So what happen in case of gymnosperm? What is there? Gymnosperms are having seep cells. So the gymnosperms, they have seep cells and albuminous cells. Means in place of companion cell, there is albuminous cell. In case of seep tube and seep tube element, there is seep cells. So seep cells are there, means they are not forming a tube-like structure. They are individual cells, but they can translocate the food material. This is important. And then there is within the phloem there are some parenchyma cells also. Say these are the parenchyma, so they are called the phloem parenchyma. All right, phloem parenchyma store food material, and also for the short distance translocation they are normal parenchyma cells. And along with this, there is an important structure is the phloem fiber. Phloem fiber, they are also known as bast fiber. Okay, the phloem fiber is also known as bast fiber. Phloem has another name, that is bast. So are much elongated sclerenchyma. phloem fiber like in xylem also. Xylem fiber is there. In phloem also phloem fiber is there. But here the sclerenchyma fiber is very long, and longest sclerenchyma fibers are found in the phloem region along with the phloem tissue. Okay, and some of the fiber in some plant, this phloem fiber are the source of commercial fiber. That thread etc. is made from the fiber, like fiber of jute, cotton, no? Jute, cotton, flax. Say commercial fiber of jute, flax, and hemp, cotton. All these are the sclerenchyma tissue. In the cotton, of course, cotton also it is a sclerenchyma, but that is not the phloem sclerenchyma, not the phloem fiber. It is a sclerenchyma. So, in case of jute, flax, and hemp, they are that they are the actually phloem fiber. The function of phloem is translocation of organic food, that is the food which is synthesized in leaf. Okay, here also like in case of xylem there is protoxylem, metaxylem. So in case of phloem also like that there is protophlloem and metaphlloem. Okay, this protophlloem and metaphloems, they are the part of primary phloem, and there is secondary phloem. Secondary phloem are formed later on in the gymnosperm, and in case of dicots only after secondary growth. Okay. Now say after this complex tissue, then higher than, as I told before, that there is a different level of organization. In the level of organization, there is a molecular level. After that, we have cell level of organization, cell organized to higher level, that is the tissue, and several tissues, that tissue system. Tissue system level of organization. Then there is organization what is called the organ level, organ and organ level, organ system, and after that comes the organism. So our next topic is the tissue system. So plants are less complex than compared to animals. Plants are simpler. So naturally, this plants, their body organization is simple. So they have only three types of tissue system. One is epidermal tissue system, fundamental tissue system, and vascular tissue system. Epidermal tissue system, that is the skin layer. Epidermis and all the structure. They say, you can say epidermis and all structure associated with or on the epidermis. Okay, that forms the epidermal tissue system. There is fundamental tissue system. Fundamental is, okay, before that let us say vascular tissue system. Vascular tissue system is made of xylem and phloem. That is the vascular bundle. Okay, these vascular bundles, they organize the vascular tissue system. And in between epidermis and vascular bundle, whatever is there in the plant, that whole thing is fundamental or ground tissue system. That is leaving the epidermis and the vascular bundle, whole thing is fundamental or ground tissue system. In the epidermal tissue system, as I told, there is epidermis, and all other structure on the epidermis. Epidermis is usually single layer. Is single layer compactly arranged cell. They are compactly arranged cells, and this is outermost layer of all the organs of the plant, whether it is root system or in the shoot system. It is in the leaves or stem or branch or fruit or flower or root branches, anywhere. This is the outermost layer. Usually it is single layer. What happened? One thing is important here. In the epidermis, on the outer side, there is a layer of cutin that is known as cuticle layer. Cuticle layer. This is the waxy layer actually. But this waxy layer, and it prevents the loss of water from the plant body. This cuticle is present in the shoot system but absent in the root. So here cuticle is absent in root, and the cuticle is the waxy thick or thin layer of chemical that is found on the outer surface only, not on the inner surface. This cuticle is also absent in hydrophytes, particularly submerged hydrophyte. Absent in submerged plants. Okay. And prevent. Another structure that what is found on the epidermis is that is hairs, different types of hairs. And the hairs on the stem, shoot system, they are known as trichomes. Trichomes only we called in case of shoot system. This can come in the form of epidermal appendages, this in the root as well, and the shoot stem, they bear different types of hair. These hairs are So trichome, this in case of shoot system, they are multicellular and may be branched or unbranched. Branched or this, but in case of root, the root hairs are unicellular elongation of epidermal cell. The root hairs are unicellular and they are the modification of epidermal cell. Of epidermal cell, epidermis of the root is also known as epiblema. Okay, so it can be said modification of epiblema cell or epidermal cells of root. And one more thing, the root hairs are present in the only in the there is a specific zone in the root, that is the root hair zone. There only we find the root hairs, not on the entire root. Okay, so there is they are limited to the root hair zone. Limited to root hair zones, and it is a temporary structure. That is, the root hairs are new root hairs are formed and the old one, but the shoot hairs, they are almost, it is permanent and all over the surface of the shoot. They prevent, trichome prevents water loss through. Another important structure that we find in the epidermal system is the stomata. Okay. See here we have a diagram of the stomata, and the diagram of stomata, this one, stomata, they are, as we know from other, we have learned that stomata, they are actually a small opening, and this is there is an opening that is known as stomatal pore, and these stomatal pores are guarded by one pair of guard cell, a special cell. These are the kidney bean bean seed shaped cells, which has on the inner wall is thick, the outer wall, that is the wall away from the pore, is thinner, and the wall towards the pore is thick. This is the guard cell. This guard cell also contain chloroplast, so they can carry out photosynthesis. The guard cell movement, by changing the turgidity of the guard cell, the stomatal pore open and close. So this way they can regulate the water loss through the transpiration. Around this guard cell, there are some other epidermal cells. Say this one, which is shown here as a green. 1, 2, 3 cells are there. 3, 4 cells are there. Their number varies from 2 to several cells. So these are called the subsidiary cells. So there are, you can say, 3 types of things. One is stomatal pore, guard cell, and the subsidiary cells. They are together forms the stomatal apparatus. Okay, stomatal apparatus consists of this stomatal pore, guard cell, and subsidiary cell. One very specific thing is that in case of grasses, this guard cells, their shape is different. They are, see, 2 guard cells here. They are dumbbell-like. They have a handle here in the middle, thinner, and 2 ball-like structure at their end. So this is dumbbell shaped. This is a exception. This is especially found in grasses. After the epidermal tissue system, there is a ground tissue system. That ground tissue system in between the epidermis, inner to epidermis, inner to epidermis, and leaving the vascular bundles, all are the ground tissue or fundamental tissue. This ground tissue, usually they are made of the simple tissue, made of simple tissue, and this simple tissue, that is parenchyma, collenchyma, and sclerenchyma. They forms the main is the parenchyma. Okay. In case of root, the ground tissue can be divided into, say, cortex, endodermis, pericycle, and conjunctive tissue, and pith. These are in case of root, fundamental tissue. In case of a stem, this fundamental tissue can be divided into, one is called hypodermis, the general cortex, endodermis, or a starch sheath, and pericycle, pith, and medullary rays. Okay, these are these fundamental tissues of the stem and root. In case of leaf, the ground tissue is called mesophyll. Ground tissue of leaves are called mesophyll, and this mesophyll, they are the parenchyma cells, and they are made of chlorenchyma, that is the chlorophyll containing parenchyma, which carry out photosynthesis. Next is that the vascular tissue system. Vascular tissue system, as I told, they are made of xylem and phloem. Here is this xylem and phloem tissue. They are arranged in a particular way. The xylem and phloem, they are arranged in a particular way, usually that is known as the vascular bundle. That veins of the leaves and within the stems and other places, those veins are made of the vascular tissue, xylem and phloem, organized into vascular bundle. The arrangement of the xylem and phloem in the root and stem, they are different. In the root, the xylem and phloem tissues, they are not found together, but they are found as a separate patches. They are found in separate patches alternately. Okay, so this is, see here, this one is xylem, this is phloem, this is xylem, this is phloem, xylem, phloem, xylem, phloem, and say this here is on this radius it is phloem, and on this radius it is xylem. Yeah, it is this is xylem radius, this one is phloem radius. So xylem and phloems are on different radius, and this type of vascular bundle is known as radial vascular bundle. Radial vascular bundle is a characteristics of root, and in the radial vascular bundle, the protoxylem is outer, that is vascular bundles, xylem is exarch. And this vascular bundles, radial vascular bundles are characteristics of root. In case of stem and the leaves, that is in the shoot system, xylem and phloem are found in the same patch. Xylem and phloems are lying together, forming the same patch. So say in this is phloem, this is xylem. They are forming one bundle. All right, and these are present on the same radius. They are present on the same radius. All right. So this is this type of, okay. পেপার গুলো টেবিলের নিচে ছিল, টেবিলের নিচে, ওই টেবিলের নিচে দেখ যা। So this is this vascular bundle where the xylem and phloems are together in one patch. They are called the conjoined vascular bundle. Vascular bundles are conjoined. So in the stems and leaf, vascular bundles are conjoined, and again ওখানেই আছে, and again this within the conjoined vascular bundle, they are collateral, bicollateral. One is called open and another is closed. Okay, so conjoined vascular bundle. Here you can see this vascular bundle, xylem and phloem is there. In between there is nothing. But in this vascular bundle, there is xylem and phloem, and in between there is a meristematic tissue, that is the cambium. This one is cambium. This type of vascular bundle is found in the dicot and gymnosperm, and this one is found in monocot plant, monocot stem. This one is in dicot stem. Okay, this one is in dicot stem. So this is called the open vascular bundle. This one in the monocot, it is called the closed vascular bundle. This is because when there is no cambium, no meristem, further growth does not take place. That is, secondary growth will not take place. So growth is closed, and in dicot stem, as there is cambium, so further growth is possible. It is open for further growth. So the vascular bundles are called open type. Say, in stem and leaves, vascular bundles are conjoined. Xylem and phloems are situated on the same radius together. In dicot, the vascular bundles are open. In monocot, it is closed. Okay, they are arranged in different way. Now next of this, so related to the vascular bundles also, usually questions are asked. They are very potent portion from where question can be asked. Type of vascular bundles and their distribution in different types of plant organs and group of plants. Next, we have to learn about our next portion. I will say about the internal structure. Internal structure of plant organ. This is particularly we will learn the primary structure. Primary structure of root, stem and leaves. So here in this we have to divide it, then monocot and dicot. That is monocot root, dicot root, monocot stem, monocot leaves and dicot leaves. Here say this is the diagram showing the roots of, diagram showing the monocot root and dicot root. This one is the dicot root. This is that A TS TS of dicot root. In the TS of, as I told that the ground tissue, here also we have to describe in the form of ground tissue. So the outermost layer is epidermis. This is also known as epiblema. Is it? This is parenchymatous, and on that the root hairs are there. I will increase the size of the diagram so that you can see it clearly. So here, this is the root here. It is clear now. Root here, and this is present on the epidermis. On the inner to the epidermis, there is a wide layer. That layer is the cortex, and inner to the cortex there is this layer. See here, this diagram is a little bit misleading. Okay, this diagram is a little bit wrong. This from the NCRT book, but this drawing is wrong here. Drawing, particularly the labeling is wrong. So actually, after the cortex, there is, I am drawing only the that stellar portion. The stellar portion here is this is endodermis. Okay, single layer. This one is single layer, and inner to the endodermis, if I so this inner layer, this is called the pericycle. This one is pericycle, and after that there is the vascular bundles. This is xylem, metaxylem, and this side on the outer side is protoxylem, and in between there is some cells like this way. Here it will be like this here also. So this is phloem, all right, xylem, phloem, and in between there is a portion called the conjunctive tissue. In dicot root, the vascular bundle is xylem and phloem patches. That is, xylem and phloem bundles are 2 to 4. This is usually, all right. And the types of vascular bundle is radial. Here in this diagram, they have actually wrongly, it has been labeled. Okay, so this is the character. In this one is monocot root. In monocot root and dicot root, there is not much difference. Only here, all other things, more or less, basic factors are same. But here, you can see this number of xylem bundles, okay, and phloems in between, their number is more. Usually, the xylem and phloem bundles are more, 6 to 8 or 6 and above, 6 or more in number. And there is in the diagram, this is the pith, this portion, pith or medulla. This is, pith is prominent. But in case of, here I told that the drawing is not proper. Here I have shown that the pith, that is the tissue in between the xylem vascular bundles, the pith portion is very less. So here the pith is small or absent. So these are the two main difference is there. Two main differences in the number of xylem and phloem bundles and the presence or absence of the pith. Okay, about one thing, that endodermis, you should know that in the endodermis there is endodermis, all the endodermal tissues or endodermal cells, they have one thing known as Casparian strips. Okay, so today I will stop here. In the next class we will continue. We have to learn about the anatomy of the stem and leaves also, and after that, based on this topic, that on the total anatomy, we will have some question we will solve. I will give you, or in between also, that what you are doing now, learning, you have the practice book or the base book or whatever. From there, you please try to solve the question. Okay, and